Oh, there we go. Thanks. So we should end up with, I guess, half the students on this one, and then we'll have another group of students at 9:30. So
Well, if the other judges don't show up, we might just get started without them, and then they can appear, when they get there. Are you aware of how many students will be attending today?
We have 10 students altogether in our category. So I see that we have, let's see, our first couple are here.
Yep, 3 students are in the room right now, and
right.
I suppose I'll turn it over to you, and you can start whenever you're ready.
Great. So good morning, students. I'm Dr. Sandy Ramirez. I'm one of the science fair judges. I haven't judged your fair before, but I usually do the big fair at the big Northwest Science thing that's at Portland State every year, except last year and this year we were virtual. So let's see. Sufyan, I think you're in the wrong category. I don't know which category you're looking for. There we go. Okay, so Brianna, you're going to be the first one. The other students get to watch, and I don't know. Let's see, Brianna, are you more comfortable if I'm watching you live with my video on, or would you prefer to just get started and do your presentation?
I can do whatever works best for you.
Okay, well, because I think I'm going to keep my video on because I know that we and you guys are missing the in-person judging, so we'll try to make it sort of realistic, and the other students will just watch in the background, and you have about 10 minutes, so you can tell us about your project, and then I'll have some questions, and hopefully our other 2 judges will hop on here shortly. And if you have any technical difficulties, Steve is our tech person. So, and Steve, I don't know if you're a teacher or what's your regular role?
Yeah, I'm actually a TOSA in the district. I work with digital curriculum.
Oh, great. Great. Well, thank you so much. So we're going to let Brianna get started so she can relax and enjoy herself. And let's hear about your project.
Okay. So hi, I'm Brianna, and my project was about the effect of pH on the rate of decrease in the mass of calcium carbonate over time. And I related the results of my experiment to the effect of ocean acidification on marine animals that have parts made out of calcium carbonate. I wanted to carry out this experiment to understand how change in ocean pH might affect these organisms, since ocean acidification is becoming a big problem. And many of the organisms that might get affected include corals, sea urchins, sea snails, and they're all keystone species in their habitats. So if the survival of these organisms is impacted by ocean acidification, then the survival of many other organisms in the same ecosystems could be affected as well. The results of my experiment found that as pH decreases or becomes more acidic, the rate of decrease in the mass of calcium carbonate increases, but as pH increases or becomes more alkaline, then the rate of decrease in calcium carbonate decreases. So I was wondering if you judges had any questions for me, if there's anything I could clarify or go in more depth about for my project.
So, thank you. And I didn't know if sometimes students want to show their slides or posters, but it's not really necessary. And I see at least one of our other judges, Tom, we went ahead and got started because we have students here, and I don't know what your area of expertise is. So if you have some specific questions for Brianna, feel free to jump in.
Okay. Yeah, I somehow ended up in the plant science group, so I discovered that, and now I'm here. So thanks for being patient with me.
Oh, sure. So in Brianna's project is a little bit about plant science because she's talking about some marine species, and I guess what I would really like is if you could talk a little bit about since I'm not like a biologist or a scientist to look at the actual what happens to them, but if you could explain that a little bit about how, what exactly happens to the carbonate parts of these.
I'm gonna try sharing my screen. Can you see it?
Yeah.
Okay. So this is some pictures of the qualitative data that I found during my experiment. And I noticed that as the pHs got more acidic, there were a lot more bubbles forming on the eggshells, whereas at the more alkaline pHs, there were less and smaller bubbles forming. So when pHs are more acidic, that means there's a higher concentration of hydrogen ions in that solution. And it's modeled in this equation here, but hydrogen ions can react with calcium carbonate to turn it into calcium ions and carbon dioxide and water. So in the picture, I think that the bubbles that formed were carbon dioxide that were forming as a result of the hydrogen ions reacting with the calcium carbonate. And since at the lower pHs there's a higher concentration of hydrogen ions, then it would make sense that larger and more bubbles form on the eggshells.
So yeah, so I thought it was interesting that you did both the qualitative, so we could see what's happening, and then can you talk a little bit about your statistical results, your statistical analysis?
Yeah. So after I got my quantitative data, after I got the data, I averaged it to find the mean rate of decrease in calcium carbonate as measured by percent decrease in hour. And from there, I also calculated standard deviations to find error bars for my graph. And after graphing the points, I used regression to create a line of best fit. And I looked specifically at the R value and the R squared value of the data. So I found that the R value for the line of best fit was negative 0.99. And the R value for a set of data can range from -1 to +1. And the closer it is to -1, the stronger it indicates a negative correlation in the data. And the closer it is to +1, then the stronger it creates a positive correlation in the data. So since the R value for the data was very close to -1, it meant that the data formed a strong negative correlation. And then I also looked at the R squared value of the data, and which is the R value squared. And this can range from 0 to 1, and the closer the R squared value is to 1, then the better fit the line of best fit is for the data. And since the R squared value for the line of best fit was 0.98, it's very far from 0, which indicates that the data is statistically significant.
Could I jump in with a quick question? I noticed you said you're using the R instead of R squared. Why did you prefer using an R value over an R squared value?
I found both, but the R value can show if the data forms a positive or negative linear correlation pattern.
Okay. That's a good answer.
Mm-hmm. So, so one of the questions that we like to ask is, you know, why you decided to study this particular project, you know, this particular topic.
So I actually have a fish tank at home, and for a very long time, we didn't really pay attention to the pH of the tank. So we were wondering why a lot of our animals were dying after we got a pH test at the store, and we found that it was like around 5, I think. So I was interested in finding out, like, because we have snails, like, what the effective pH, what kind of effect pH might have, um, on the animals in there. And I thought I could connect it to this bigger topic of ocean acidification, while I'm at it.
So
Great.
how,
And go
how
ahead,
did
Tom.
how did you measure the pH?
Um, like in the experiment, or like a
Yeah, you mentioned. I think. Could you clarify that?
yeah, I wrote about it in my procedure. So in order to manipulate the pHs of the solutions, I added pH buffers to 100 milliliters of water. Um, and after that, I also used a pH sensor to measure the pH, since the pH buffers can only make the water relatively close to the values.
Okay, so it's just a probe you stick in the water and it has a digital readout. And where did you get that?
I borrowed it from school.
Okay, that's good.
Great. And we have our third judge joined us also. So we went ahead. Sorry, we started without you because we wanted the students to not have to
Right.
wait.
Yeah. Sorry, I didn't have the Zoom links. I don't know. You guys must have found them somehow. They weren't in the spreadsheet where they said they were going to be. So yeah, I'm sorry I'm late here, but so I have a question too. I assume this is the eggshell one, right? The calcium carbonate.
Yeah.
Yes.
Yeah. Okay. So you only had 100 mils of buffer, and you said that was barely covering the eggs. How do you think that similar to what you'd find in the ocean, where you've got an unlimited supply of acid and whatever's going to be in there? You only got 100 mils. You probably use that up pretty quickly, and then the pH probably changed. Did you think about maybe measuring the pH at the end and see how it might have changed? Oh, you did. You did measure the pH, right? That's I'm thinking of something else. So you did measure the pH, but how do you think that might compare to the ocean, where you've got an unlimited amount of acid as opposed to just a small amount?
It would increase the effects that I saw in the experiment, since um the shells in the ocean would be completely covered by water. And then since there's a lot more, since the pH would be higher, since ocean acidification has greatest effects near like the surface of the ocean, which is where a lot of the carbon dioxide comes into contact with the water, um, then I think that the effects that I saw in this experiment would be a lot worse for
Right.
if it was in the ocean for real.
Very good. Thank you. Yeah, because your pH was probably changing quite a bit as the acid was being used up, and you only had a limited amount there. Also, you didn't rinse the protein out of all that. You probably had a lot of protein left inside the eggs. And you mentioned that yeah, it's probably hard to get them dry with just a little pinhole. But you think about maybe you got a lot of protein left in there. The protein is going to be reacting with the acid too. What do you think that might do?
Then it would also cause the masses of the eggshells to decrease a lot more.
I know. So you probably had to blow out all the egg to get the weight down, so your percentage didn't make much difference as opposed to putting in a whole egg with all that weight, extra weight. Yeah.
Did you eat the eggs?
Like I don't know. I chose to use eggs for this experiment since I wasn't really able to find shells because of the pandemic, and I thought that eggshells would work as an alternative, since we have a lot at home and my family was going to eat them anyways.
Okay.
Great. So, so Brianna, you can stop sharing your screen, and we'll see if we. So we're probably close to the end of our time. I didn't know if the other judges had any more questions for you.
I don't.
Great. Great. Well, Brianna, thank you very much. Hope you'll stick around and listen to some of the other projects, and really great job. Thank you so much for participating.
And thank you for your
Thank you.
feedback and your time.
Great. Great. So I'm not sure how to pronounce this correctly. Pooja, is it Pooja?
Yeah.
Great. So Pooja, welcome to the Science Fair, and we would like to hear about your project. And we have about 10 minutes. So give us the kind of the overview of your project, and then the other two judges and I will have some questions for you.
Okay. So just a brief overview of the project. So the research question that I was
And
answering.
I'm sorry, you might want to turn your volume up or be a little closer to your speaker.
Can you guys hear me now?
That's a little better, Phoebe.
Okay. Can you guys hear me now?
Yeah, I think that's good. Just try to speak up a little bit. That's good.
Okay. So the research question that I was attempting to answer was, to what extent does temperature affect the density of cyanobacteria in Lake Erie from July to November between 2017 and 2018? So my motivation for this project came from actually completing a different research paper on the effects of urbanization on water quality in Nepal. And I really wanted to kind of go more specific into the actual effects that different things have on water quality. And one of those is cyanobacteria. So basically, this is an issue in the US as well, specifically in the Great Lakes. So as I outlined in my research paper, there have been multiple instances in which cyanobacteria has either been fatal for humans or animals, more specifically fish, because it deoxygenates the water, and so it's extremely harmful for ecosystems. But then it's also harmful for economic activity in areas. So for example, in 2014 in Toledo, Ohio, over 400,000 people were left without usable water for over the weekend. And so this is going to be an extremely increasing problem as the effects of climate change exacerbate, because we're going to be seeing increased eutrophication, especially through more urbanization near these waterways. And so a couple of ways that we can adapt to climate change related disasters is first by preventing the disaster from happening in the first place. So in terms of cyanobacteria, there are multiple ways in doing this. One of them is riparian zones, basically creating a buffer between the water ecosystem and other nutrients that may flow into it. So this is something that we see in the Midwest because of the rural communities. Another way to adapt to it, climate change related disasters, is just to mitigate. And then the third way is to adapt. So I was really focusing on the adapt aspect of this, because if we know the temperature in which cyanobacteria flourishes, then we're able to kind of keep our communities more informed and keep them safer. So in the end, after completing this research project, I found that the optimal temperature for cyanobacterial growth in Lake Erie specifically was between 66 to 77 degrees Fahrenheit. And so this is useful information for people in that area so that we can be more aware and prepared to protect our communities against the harmful effects of cyanobacteria when the water temperature reaches that range. And I'm open for any questions.
So that temperature range, is that most of the summer?
So the way that the data was collected was only between July to November, because that's when the most amount of data by NOAA was collected. And so we're seeing that happening closer to the end of summer, so in the July, August months.
I think you mentioned two other factors that could have affected the growth. Can you remind us of what those were?
Yeah, so one of them is the nutrient concentration. So as I mentioned before, eutrophication is a huge issue for cyanobacterial growth, and that kind of connects with increasing patterns of urbanization. So what happens is that the nutrients enter these waterways and they're able to, like the cyanobacteria is able to feed off of these nutrients and they're able to grow in population. And they're also able to deprive the ecosystems of these nutrients that are necessary for their survival. So that is definitely one of the effects that I would like to study in the future, the effect that nutrients also have on cyanobacterial growth, because then I could control for that variable.
And I think you also mentioned sunlight. So what would be a fairly quick way of looking at the data and trying to get a handle on how important those are?
Yeah, I think conducting 2 different experiments. So like with the sunlight, conducting an experiment with UV light, or the UV index versus the cyanobacterial growth, and then for the nutrient concentrations versus cyanobacterial growth, and then really comparing the trends that we're seeing. And if those are similar trends, then we would see that those probably have an influence on the cyanobacterial growth as well.
Okay. You also might just do a time series of your data across the year, because if you know the month, you basically know how much sunlight there is. And typically nutrients, they get mixed up into the water in winter and then often used up when you get the blooms. So if it with the temperature, so you started in July. So were you starting kind of at the peak temperature? Because usually the temperature, again, it's cold in winter and slowly increases over as the summer goes on. So if you compare so you might get the same temperature in, say, June and September. And if you compare the growth on those two, that would be an interesting piece of information.
Yeah, I think the only area that's limited in is just the availability of data. But if we were able to get like data for June, in terms of the data was available for the water temperatures, but not for the cyanobacterial growth. So I think that was the limiting factor on this experiment.
Oh, that's right, because she wasn't gathering her own. She was just getting the data that was provided by NOAA. Yeah.
So Pooja, here in Oregon, we have the rivers, and they, periodically, based on the weather, like if we have a lot of rain and storm drains overflow, that affects the water quality in the Willamette River, say, or the Columbia. But I was wondering, at the beginning, you were talking about industrial effects, but also there are the agricultural effects. And I'm not sure if I remember whether you talked about, which one is worse, in terms of affecting the water quality and leading to the bacterial growth. I know you were looking at temperature for this, but I know you were also talking about urbanization and that type of thing. So.
Mm-hmm. It really depends on what preventive measures we have. So as I talked about at the beginning, we can use like riparian zones in order to create a buffer between the nutrients that might be coming in and water ecosystems. So if we're able to prevent the effects of urbanization from having too much effect on cyanobacterial growth and by using riparian zones, then urbanization will have less of effect. But it really depends on kind of the preventive measures that we're taking, because this is sort of a very real life experiment in that there are a lot of different variables out there. It would just depend on how we're preventing nutrients coming into the ecosystem.
So you only had temperature measurement in one place, but you were measuring the algal bloom all across the whole lake area, right? Do you have any other data on temperature, how much the temperature varies around the lake?
Yeah, so when I looked at the cyanobacterial density maps, a lot of the cyanobacteria was concentrated on the west side of the lake. And so the NOAA has different places around the lake where they collect for the temperature. And so I specifically looked at the spot where they collected temperature on the west side of the lake, since that's where the most amount of cyanobacteria was concentrated.
So you were measuring temperature in the area where there's the highest growth?
Yes.
Okay. Okay. But from looking at the maps on the different days, it looked like for some certain areas where it had a lot of variation over time. So I just wondering if your data would be the same if you just focused on the temperature and the algal growth right in that specific one area as the temperature of that, rather than, because I don't know how much temperature variation there is around the lake and how much the algae tends to move around, or if it tends to stay put. But you know, your data for how much algae was for the whole area, and I don't know how much your temperature varied around that lake as well. Why there's different amounts of growth in some areas and some not in others. Maybe there's just more, like you're saying, more nutrients coming in from farms or whatever. But
Yeah, I think one of the observations I made was usually there's more cyanobacterial growth on the sides of the lake. And so that's most likely because there's more urbanization there and nutrients are able to flow more easily there.
coming in, yeah.
Great. Well, Pooja, thank you very much. I think we're about out of time. I think we only get about 10 minutes per student, but this is a very interesting project and very timely. So unless one of the other judges has a burning question, you know, we can tell you thank you.
Jump in real quick. Do you know what depth the temperature was measured at?
Oh, you're muted. Can't hear you.
Oh, well. Because I think the lake does set up pretty strong stratification in summer, but I'm not sure.
Great. Okay, well, thank you. Thank you very much, Pooja. Good job. And stick around and listen to some of our other projects. Thank you so much. Let's see. The next student that I have is Olivia, but I don't see her on here. So we do have, is it Eshal or Eshal? Are you able to get your video showing? There you go. Good morning.
Hello.
So Eshal, you have about 10 minutes to give us an overview of your project and you know, maybe spend a few minutes and then we can go ahead and have some questions for you.
Okay. Hi, my name is Eshal, and my project tests the relationship between the concentration of motor oil used to germinate mung beans and their growth over time. I hypothesized that if different levels of motor oil from 0% to 10% were distributed to mung beans, the germination of plants given the most oil would be the slowest and least out of the varying amounts. This is because toxic oils had previously impacted animal species and land negatively, which means it may contain harmful chemicals that can damage scientific processes occurring within the mung beans. To test my hypothesis, mung beans were exposed to different concentrations of motor oil over the course of 7 days at 0%, 1%, 2.5%, 5%, and 10%. Every 24 hours, the beans were rinsed, measured with a ruler, and re-soaked with water and oil. The results from the experiment partly supported the hypothesis that mung beans germinated with 10% motor oil would grow slowest and the least. For instance, the final length of the mung beans given 10% oil was equal to that of the plants given 5% oil. In addition, the growth of the mung beans given 10% oil was not constantly slower than the growth of mung beans exposed to less motor oil. However, a correlation between motor oil concentration and mung bean growth was still identified, as the control group of 0% motor oil grew far faster than all the experimental groups. Multiple sources of error existed in my project, including a flaw in my design and the measuring tools used. A systematic error in my project was rinsing all the mung beans before starting to measure. This caused the mung beans with the most oil to be dry the longest, which may have made them more brittle. To improve this, time of soaking could be added to the listed constant. From the project, it was found that motor oil has a negative effect on mung beans. This data is vital as it works towards creating change for the benefit of Earth and its inhabitants. Thank you. I'm open for questions.
Thank you so much, Eshal. So I'm going to let my scientist judges ask you the hard questions.
So as you know, I think you noted oil and water don't mix, right? So the seeds so where was the oil in relation to the water during this experiment?
Container had the beans and then the water, and oil kind of created a surface on the top.
Okay.
And it was one of my sources of error, and I think I could have improved it by adding an experimental group with only oil so that it touched the beans directly.
Okay, so the seeds were down were down in the water as opposed to I mean the most of the contact was with water. Then do you think much? So why do you think there was a difference then if the oil wasn't touching the plants?
I think it would have slightly made their growth like better than it would in. I think it improved their growth more, and I think that in the natural environment that does happen because there are oil spills that are leaked into the oceans that affect animal species.
Well, in the natural environment, they'd probably be in the ground and it'd be oil maybe getting into the soil, probably where your beans would be most likely, right? You wouldn't have the problem with it floating because it would be mixed in the soil.
I have previously done an experiment with mung beans, and it was something I knew how to grow, so that's why I chose it. But I think I could have switched to a different plant and grown it in soil so that it could have been more accurate.
What do you think would have happened if you mixed in a little surfactant, which generally reduces the surface tension and allows oil and the water to mix? It's kind of probably the easiest way to get some is stick in a little dish detergent.
They're on mute.
We still can't hear you.
Still muted.
Is your computer on? Is your computer so you're?
Yeah, she is not muted and her mic is on, so there could be a technical issue with Zoom here. So, Ishal, you may need to type your response.
Can you hear us? Can you wave at us if you can hear us? Okay, that's good. Well, I'll ask hard questions and Brian will answer them for you, and that way you'll come off.
Yeah.
You'll probably win the contest with doing that.
So I actually had a question too. It's like, so I could certainly see where motor oil would be icky and have negative effects on any, I mean, I certainly wouldn't want to eat anything that had been in motor oil, but I was wondering if it's the fact that it's oil, and I was wondering that, like, if you did the same experiment and had motor oil for some and then used something like vegetable oil or, you know, some other oil type of product that was, you know, edible, say, for example, whether it would just be, so I don't know about the chemical composition and how that would be affecting the growth of your mung beans, but had you thought about that? Right, right.
Hi, my name is Isam Qureshi, and my research tests the correlation between the amount of level in certain communities and that grows over time. To test this inquiry, many scientists.
Whoops.
Sorry, that was me.
Oh, that was her. That was her video.
Yeah, I was looking something up.
So, one question I had. So you did multiples of each oil. I didn't have your information right in front of me. But I don't believe you had any statistical analysis of how much variation there was between the different samples. Did you look at that at all? That's not what I want either.
It just seems like you've been getting a lot of hard questions here, but it just kind of an overall comment is that it seemed like you did a pretty nice job of kind of presenting your data and trying to think about what could cause the differences in it. So you're getting hard questions here, but I think try to take it positive, because there's a lot of really good stuff in what you did, and I was pretty impressed with it.
Yeah, I know that's a really big problem. I mean, you see people, dumping antifreeze and oil and stuff down their storm drains after they change their oil, and that stuff goes right out into the environment. They just think if it's dumped in the water, it's going to be gone, but it's still there, getting into the soil and into the food we eat and everywhere else. So this is a really important thing to know what you're studying.
Yeah, you did a lot of good work on this, and one of the questions that we like to ask is if there's something, what it is that you're hoping to learn from all this, or where you're going to go with it. If you're interested in pursuing, like environmental accidents and misuses and all of that going forward. We don't need to rush because we don't seem to have any other students coming. So just, take your time.
Oh, that's right. We have two different groups, don't we? We have another group later. Yeah.
Yeah. Yeah, Steve, I don't know if there's any way to find out if there are students that went to the wrong groups that are hanging out somewhere else.
I think if they were in the wrong room, they would have shown up by now.
Oh, great.
So what sorts of things does your environment club do?
And, while she's typing, Pooja and Brianna, if you have any questions or if you have any comments, it's interesting. All three of your projects are, I mean, I realize that's the category we're in, but all three of your projects looked at similar kinds of things too. I think, yeah, so Brianna and Pooja are seniors, but Ishal, you're just a freshman, so we could see you back here next year doing an even more exciting project.
Maybe see you in person there.
Yeah, that would be great. So yeah, so Pooja and Brianna, if you had any questions, you could ask them also.
I was just looking at your data. It's kind of interesting that the motor oil makes the sprouts smaller, but the length of time, everything sprouts on day 4. So it's motor oil, it doesn't not make them sprout, and it doesn't make them sprout sooner or later. It just makes them sprout smaller.
Yeah, they don't grow as fast. Yeah.
And I think in the discussion there, it did kind of mention that it would be interesting to know actually why the oil is having the effect it's having.
Especially when it's not really in contact. It must be something dissolving in the water.
Well, there is going to be a certain amount. I would think that if you have it floating on the surface, the water just sees a layer of oil up there. If you put more percentage, you're probably just going to get a thicker layer, but it's hard to see how that would really, the water is still seeing oil up there, and it probably doesn't really notice how thick the oil is, which is probably kind of consistent with the results. I mean, it sort of has effect in the 5% and the 10%. Have very similar results, and that would be consistent with that.
Yeah, I can't recall if the seeds were already in the water and then the oil was put on top, and then they wouldn't have had contact. But if they were dropped through the oil, they would have that momentary contact, as they were, they get oil on them. That's the first thing they would hit when they were dropped in.
Yeah, I mean, oil and water don't mix. I'm not sure how, I guess bean and oil would have the same, or are beans is whatever hydrophilic as what water would be.
Yeah.
A lot of organic material, I believe, soaks up oil pretty readily.
Yeah. Like I said, there are probably water soluble things in the oil that might be coming into the water as it's sitting on top. That's like a lot of good projects you guys do in your club. That's good to have concern in a group and work together.
Great. Well, ladies, I think that you have been hard at work this year, it sounds like, with your projects, and I'm always glad to see people participate in science fairs, and you all did a super duper job. And I think we're going to let you go so that you can relax and go get some breakfast or whatever. And thank you so much for participating in the science fair. And if the other judges want to stick around for a minute, we can chat before we take a break. So thank you very much.
Thank you.
Thank you.
Thank you.
Great. So, hi guys.
Hi.
Hello.
Yeah. So as you can tell, I'm not a marine biologist or anything related to the environmental sciences. I'm actually a psychologist. So usually I judge the behavioral sciences category, but it looked like they had a full crew in that. And I usually don't judge your fair either. I do Portland and the Aardvark one, and then the final one. But this is interesting. And of course, I'm very interested in the environment. We're all Oregonians, right? So we care about the planet. And it sounds like, I'm disappointed that we didn't get our other couple of students that should have been in this first group. But I didn't know if you had any comments about any of the projects or anything we should be keeping an eye out for in our next round. I saw that none of you could do the finalist judging, and I'm not really sure why I had clicked that by mistake when I was signing up, because I have a conference that starts here. I mean, it's a virtual conference, a national one. It starts around noon. So I'm gonna, after we're done making our choices and sending them on later this morning, then I won't be able to be back to do the finalist judging. And I don't know enough about the sciences of the other categories to to ask the good questions anyway. So
So what's the, I know they got the schedule here somewhere. Where did that go?
I think the second group starts at 9:30, and then we have until about 11. Well, if we have five, or it sounds like we have an hour and a half or something.
Have the other, all the students have left this meeting, right? They're not.
Yeah, there are no students on here right now. Just
Okay.
Steve is our tech host.
We have somebody just joined us.
Olivia
Oh.
just joined us.
Oh, well, great. Well, then, good. Well, we have time to interview Olivia then.
She was here.
So which were the numbers of the ones that haven't showed at this point that should be here?
So I don't know if we were just having the first five, we should have gone through. So Olivia is 16, and then we have one more that's VE0018, if that person shows up. Olivia, hi. I'm Dr. Ramirez. I'm one of the judges for your category, and you're the last one. Hi. So we would like to hear about your project for a few minutes. Sorry, I'm losing my voice. Hear about your project for a few minutes, and then the other judges and I will have some questions for you.
Okay. Am I too late or am I on time?
You're fine.
Okay.
You're great.
Okay. Hi, my name is Olivia Chisire. I did my project on gray water. And gray water is a type of water that is commercial and residential use. It does not have any fecal contamination in it. And my question was, does gray water affect the growth of green onions? The null hypothesis was that the different types of gray water did not have any effect on the green onions. And when I tested my hypothesis, there were 3 different independent variables of the gray water, which consisted of hand washing dishes water, eco-friendly laundry detergent, and non-eco-friendly laundry detergent. And I had a control group of filtered water. The project was performed by submerging the roots of the gray water in submerging the roots of the green onions in the gray water for 14 days and collecting the results at the end and at the beginning. The reason why I used a hydroponic system of submerging the roots instead of putting it in soil is because this project is meant to reuse and recycle water, and hydroponics is saves 10% more water than if you were to plant in soil. And so during the process of 14 days, they lived under a grow light and were closely monitored. The end heights showed that the null hypothesis was rejected because the not all the heights of the plants were the same. And the eco-friendly laundry detergent had the highest growth rate average, following with the filtered water, which also followed the hand washing dishes water, and the group with the least growth average was the non-eco-friendly laundry detergent. This rejected my null hypothesis and accepted my alternative hypothesis. I did this project because I'm interested in helping our environment and ways at home where you can do something yourself and make a difference and not just wait for other people to do something. Thank you.
Great job, Olivia. So I had a question when I was reading your project that, and I might not have understood exactly how it was, but it looked like you had a whole chart that was like a hypothetical set of results, and I didn't understand the purpose of that because you said something was made up.
Yes, okay, there was 2 graphs in total. The first graph was the bar graph, which was to show the difference in growth. And the ANOVA test, which is what I think you're referencing, is to test if my data rejects or accepts the hypothesis, and what basically is explaining what my data actually means. And what it's saying is that it's showing the difference in height. So if they were all at the same level, then my null hypothesis would be supported. It's just going the deeper layer and understanding what my actual data was representing.
Right, but I guess there was something where you said number 14. So you had a whole, it's like you had a row that was your actual plants, and then you had something that was row 14 that you said was made up. So was that, I mean, you used the term made up, and is that what you were talking about for your null hypothesis? I guess I'm just not understanding it very well. Maybe one of our science guys understands and can clarify that, but I didn't quite understand what that was.
I'm trying to find where that's mentioned here.
I just see 2 graphs. One is, again, the bar graph, and next to that is just kind of the ANOVA, which I think is pretty similar to error bars on the results.
Right.
So how did you decide to use the statistical approach that you used? Or where did you learn your statistics?
I learned how to do the one-way ANOVA test from my mom, who is a mathematician, actuary, and Khan Academy.
Wow.
Okay.
That's good.
Yeah, I wish somebody who was a statistician would adopt me. You didn't actually show your individual data points, correct?
Correct.
Okay.
Because I took the averages off of each group.
Is another question. So you had 4 or 5 point trials. So one bar graph is the average of how many data points?
It's the average of 7 data points.
7 data points. Okay. Did you used the average? Did you think about using the median?
No, I did not.
Okay. What would be the advantage of using a median? Do you remember what the median is?
Yes, I do.
Okay.
I thought it would be more fit to use the average instead of the median because it just got a more accurate reading, because there's some outliers in the data and I wanted it to be even all around.
So would an average or a median be more affected by outliers?
Average would be more affected, sorry.
Okay.
So I'm curious about how you actually got your solutions, like the dishwashing solution. How did you come up with your your water for that?
Okay, so we do the dishes in our house every single night, and so I just took a sample of that and put it in because the water has to be cycled out every 24 hours, or else the um, nutrients from the gray water will contaminate the plants and it will start stinking. And so I just change it out every 24 hours with the water that is from the original source.
Oh, I see. So your dishwater could be different every day then, right? Because
Yes.
depending on what you were washing and how much soap you used and what kind of oil, how much oil got into it and everything
Yes,
else.
I wanted
Okay,
it
so
to
that
be as authentic as possible.
okay. So so all these solutions you had to make them up. You had to get different water every day then.
Yes.
Okay.
Because I was referencing if somebody was doing this in their actual household and not in a lab or something. So if somebody wanted to do this, use this to grow, I don't know, a house plant or something, they could have the best results in everyday life.
So the laundry detergent, I can't remember if you mentioned it in here. I don't let's see it right away, but so you put a certain amount of laundry detergent in a certain amount of water and then mix that together, and that's you would do that fresh every day.
Yes.
Okay. So was the
So
water pretty sudsy or when you used it or?
No, in on the back of the laundry detergent that I used, which was a Kirkland, there was a reference for how much you should be using every single day, and then I took those measurements down to my experiment, and it was 1 cup equals 1.5 teaspoons of laundry detergent, and that was for the eco-friendly laundry detergent. And for the non-eco-friendly laundry detergent, it was 1 cup of water to 1 teaspoon of
Okay, so they didn't all get the same amount of necessarily of how much detergent. It was all based on how much you would use for a wash load of clothes, sort of
yes.
then. Okay. And then, all right, so that's fairly concentrated. I mean, the amount of soap you'd have in water as you were washing clothes, when they actually get out in the environment, did you think they would be that concentrated?
Can you please repeat the question?
So you have a fairly concentrated amount of detergent in your water when you're actually washing the clothes. But when that gets out into the environment where the plants would be, is it that concentrated, or would it be? You think it'd be that the soap would be that concentrated when they actually get to the plants?
No, I think it would be just as concentrated because um, this experiment is for if you were having a gray water tank at your house, and it wouldn't be used in the sense of like
Oh, I see.
supply.
All right, right. So, right. I'm sorry. So, yeah, you're taking this water and using it directly for growing your plants in, right? So it wouldn't be okay. Well, even then, though, you'd probably it would get diluted a fair amount because you're going to be rinsing and whatever after you wash your clothes. So you'd be probably diluting it somewhat after that, but you'd still be a fair amount of soap, right? I was thinking, yeah, this was just getting dumped down a drain and getting out in the environment. But, you're actually reusing the water directly. So yeah, you're right,
Yes.
actually.
Yeah.
Thank you.
So would you be willing to eat onions that were grown in gray water?
No, I would not eat. That was if I was to continue this project, I would see the actual effect on if it was edible or not. But was just a test group, and it was the most hardy plant that could sustain this kind of damage. I also, in my air analysis, I tried to grow a different type of plant from a nursery, but it did not have any roots connected to every single different leaf that I was trying to test, and this was the easiest option.
So ornamental flowers might be a better use for gray water.
Yeah.
Yes.
So the eco-friendly detergent behaved about the same as pure water in terms of the growth. So the nutrients in from the detergent doesn't look like they're really helping the plant. It's just, it doesn't hurt it. So that's.
Yeah. I was very surprised by that.
Yeah, I think mostly detergents have, you know, phosphates and things like that that end up somewhat being a fertilizer. Like in one of our other presentations, you know, they're worried about things getting into the water in a lake, and a lot of those things would be detergents and things like that. You know, phosphorus and things like that, that actually would help plant growth. But
Yes.
I would think at some point it's going to be a negative effect, like what you've seen if it's too concentrated.
Yes.
Probably the non-eco-friendly actually has more nutrients in it than the eco-friendly.
Right. Yeah. So why do you think, what's the difference between the, do you know what the eco and non-eco friendly detergents, what the difference is?
The eco-friendly laundry detergent does not have as many of the harmful chemicals, and it is more concentrated, as you said. And would you like the exact chemicals, or would you just like an overview?
Yeah, I just curious if you what do you think would be, I mean, you're not a chemist, but there's something in there that's causing some harm, and do you, if you knew the difference between the two things, yeah, you might know what's actually causing the problem.
Yes. So, in the all non-eco-friendly laundry detergent that I used, there was the biggest difference was microplastics, and harming the microplastics, harming the environment, and this was found in this laundry detergent and from past articles and research. But the eco-friendly laundry detergent does not have this.
Mm-hmm. Probably perfumes and things too, and dyes and things. All those things can all have an effect.
Yes.
Yeah, probably whatever they use for fillers, or, you know, to make it cheaper and go further, or who knows what could be in there. But well, Olivia, great job. Thank you so much for coming with your project today. And I think we're gonna let you be free. And thanks for participating in the science fair. And since you're a freshman, we expect to see you back next year with an even more exciting project.
Yes.
And even more exciting statistics too. Get on your mom about that.
And your original question, I think you were referencing the day 14, and that just meant that I collected the data on day 1, which was a controlled variable, and day 14, which was the different data, if that helps at all. So.
Great. Okay. Thank you so much.
Thank you.
Thank you.
Okay, do we have any more last minute contestants?
See, we're missing one, right?
Yeah. It's the, let's
Flood
see,
control.
effective flood, yeah, flood control thing.
Oh,
So
yeah, I was hoping to talk to him.
Yeah, his project was sort of incomplete in the submitted material, at least what I had. Was it the same for you guys?
Yeah, I mean, it would have been nice to have more pictures and of actually what was being done there. I couldn't really tell where the water was being poured and measured and all that sort of thing, but from what he had. But.
Great. So when you guys were reading these, did you complete any of the scoring stuff?
Yeah, we both filled in the spreadsheet with some scores.
Great, great. So I did whatever was in the online thing. So so when we're done later this morning, we'll have our, we can go ahead and talk about the projects. And meanwhile, it looks like we're